Today I'm going to be talking about the vision of the L2S2 platform. I'm going to talk about some strategic features that went into the design and give you a brief introduction to the development and implementation. And then we had the privilege of being able to show this audience a video reveal of the Lilly Life Sciences studio for the very first time. Later today it will actually be made public, so this group's going to get to see it first. After the video, Todd DiCarlo is going to come up and speak on the scientific and technical details. So he'll be taking a deeper dive into the functionality of the Lilly Life Sciences Studio, the rationale for why we chose to do what we did. And then I'd like to ask everyone to hold their questions until the end. The last 20 or 30 minutes of the presentation today, we're going to ask others from the Lilly Life Sciences Studio design team, which are seated up here, to actually participate in more of a roundtable Q&A. And at that time, we'd like to open it up to any questions that the audience may have. So if you have some good ones during the talk, please take a note and save those for the end. So let's jump right into the Lilly Life Sciences Studio. The vision for this platform was really all around revolutionizing the access to molecules. We wanted to streamline the design, make, test, and analyze drug discovery cycle that many of us as scientists participate in every day. Essentially, we wanted to open this platform up to researchers all around the world that had ideas or hypotheses. We wanted those researchers to have a collaborative remote portal where they could actually log in and have access to the latest chemoinformatics, bioinformatics tools, which would allow them to refine their hypothesis. We wanted them to be able to assess the latest and greatest retrosynthetic analysis and have an opportunity to do reagent or building block search and selection in order to further refine that hypothesis. At that point, we wanted the researcher with a refined hypothesis to then be able to submit that experiment or order to an automated platform. And so that experiment needed to be translated into a format that could be understood at the automation layer. Once that experiment came onto the system, we wanted to be able to quickly synthesize, purify, and test those molecules. We then wanted to be able to immediately get data on the molecule or the compound of interest that could be fed back in to refine or further refine or iterate upon that hypothesis. One of the key paradigm here is this idea to data. We wanted to go from an idea in a researcher's mind to data as quickly as possible so that we could then iterate and come up with a better idea. As we set out to build this system, we really had 4 design and sustainability goals that we wanted to put into the design of the platform. We wanted this system to be adaptable not only for Lilly's needs, but we really wanted it to be adaptable for the needs of other scientific and research organizations around the world. We also wanted a platform that would continue to evolve and not only be current, but be state-of-the-art for the next 20 years. And so we came up with these design and sustainability goals that were known as the 4 R's. The first one being we wanted a platform that was reproducible. So there's no better way of doing that than with automation. We wanted something that was robust. We wanted something that was not only reproducible in function, but we wanted to be able to, if we made a molecule, we wanted to be able to remake that molecule with the same yield and purity as the first time. We also wanted to make the time period reproducible. So if it was going to take 5 hours, we wanted it to take 5 hours the next time. Something that we found is very that varies when we have manual operation. So we made the decision that we wanted to make this as automation fully automated as possible. We also wanted a platform that was responsible, and so you're going to hear today over the course of this talk this idea of just-in-time model. We wanted the ability to use building blocks and materials to make these compounds just in time. We also wanted to make compounds that could be tested just in time. The scientific industry spends a great deal of time and resources and money saving compounds that very rarely get used. We wanted to be able to make a compound, and if it looked promising, we could quickly remake it and use it again. So we're trying to be responsible stewards to the environment. We also wanted to build a platform that was relationship building. And so part of that was building this platform in a innovation hub, or a location where we'd have access to building those scientific and technology relationships. And I'll talk more about the location that we picked for that. We also wanted it to be a platform that we were not only opening up to other scientific organizations, but relationships when it came to the technology vendors that we were working with, the kit manufacturers, and then, of course, other public and private scientific organizations. And then finally, we wanted a system that was rewarding. So we built this system not just for the scientists that are working today, but we really built this system with that high school boy or girl that's sitting in a chemistry or biology class today, having aspirations of what they might do, of the ability to make a life-changing or a life-saving medicine someday. We built this system with them in mind, and with that, we wanted it to be future-proof. So the location that we chose for the Lilly Life Sciences studio was at our Lilly Biotechnology Center in San Diego, California. We did this for many reasons, but one of those was we wanted it to be a relationship building in an innovation hub. We had a unique opportunity with the build-out of L2S2, and that was at the same time that we were building L2S2, we were also doing a Lilly Biotechnology site expansion. And so this started in July of 2015. And what you're seeing in the picture here is actually what used to be the Qualcomm building. And we took that over in July of 2015. The next picture shows what it looked like later that year in January, where we pretty much took it down to the studs and then began to rebuild it in the manner that was fitting to Lilly. And so by December of 2016, so nearly a year and a half, we went from the original building to the new Lilly Biotechnology Center. Now, during that time, we were also planning out the Lilly Life Sciences studio, and you can imagine the opportunity that comes with that, but also the challenges that come with that when you're trying to build something when there's also not a foundation there. And so one of the challenges that we had was just thinking about the space of evolution. And so the original plan for the Lilly Biotechnology Center for the site coordinators, or the people that thought about this, was this idea that if we wanted to go fast and we wanted to move through the process, it would be great to have different functions of the drug discovery process co-located together. And so this early rendering of one of the original site maps actually shows a next-generation automated synthesis lab. Lilly is a pioneer and has been for several years in automated synthesis. There was an original idea that we build a next generation as part of this, and then having co-located very close to that system would be global sample management, analytical technologies, medicinal chemistry. We'd have NMR capabilities. And so everything would be very co-located, same floor of the building. And that would essentially help the collaboration speed up. But as we assessed what was happening in Indianapolis and other research areas, we had automated many of these functions before, and we had automated them very, very well on their own. So we have automated global sample management, we had automated synthesis, we had automated biological assays. But what we found as we assessed this process is that we could speed things up locally and in one certain area, but it really didn't move the radar or the needle on the speed of the entire drug discovery process. So the overall process was rather inefficient. And what we found is we needed a way to better connect these functions, to kind of de-silo these different areas. And so one of the things, as we began to think about, why don't we knock down the walls of these different areas and put everyone physically into the same lab space, we uncovered that there was a large structural diagonal beam for an earthquake beam, essentially, that made it very, very challenging to have one big open space that could house everyone. So what you're seeing here in this diagram is actually where this beam was located, and one of the things you'll notice in one of these second drawings of the lab space is that we took away a lot of the walls between global sample management, analytical technologies, medicinal chemistry, and it began to be more open floor plan. But as I mentioned, just being close in proximity wasn't enough. One of the things we assessed is that some of our longest points were taking from one platform to another or from one lab to another where it relied on humans to actually walk those over. People would be on lunch breaks, people would leave at 4 or 5 in the afternoon, we had holidays, and so a lot of times it'd be hurry up and wait for samples to sit in one location until on a Friday afternoon until Monday. And so, again, going back to we wanted something that was reproducible. We wanted it reproducible not only in yield and purity, but reproducible in the time that we'd expect to get answers. And so you then see in this next drawing that we began to think about how do we bring all these functions together in an automated way where there would be some level of connectivity. And the next diagram shows the idea that we would actually physically connect these different areas of the scientific process using some sort of track system. Now, in addition to physical connectivity, another important part is the digital or the virtual connectivity. Many of areas across the research organizations, not only at Lilly and elsewhere, are very different. When you go from sample management to biology, there's different LIMS systems, there's different barcode tracking capabilities. Some areas have them, some don't. And so you're always fighting not only the physical transport of samples, but also the digital transport of sample information. And so in paralleling the physical connectivity, we were also thinking about how do we connect this from a digital transformation as well. And so this is one of the original drawings, once we had kind of specced out our space, that actually shows a fork-shaped track system. And so the idea would be this track system would be the physical connection of moving samples through that entire drug discovery process. This is now a top view 3D rendering of that same fork system. But originally we had about 3,800 square feet that was spec'd out for this. By the time this was said and done, we had 11,500 square feet of open lab space with connected modules. We partnered with the Morse Group, and the Morse Group, for the most part at the time, was very well known in other industries, but in lab automation they weren't known quite so well. But we partnered with the Morse Group knowing that we needed the flexibility to build a system that was the scale of a manufacturing lab, but we needed the flexibility required in research. And so the Morse Group, under the Morse Group umbrella, there is a mechanical engineering company. There's Automation Solutions of America that they do custom robotics. They have an audio-video company that does a lot of the high-end video equipment on the Vegas Strip. And they also have a safety company. All of the things we needed that if we didn't know what it was going to take from a utilities and a power standpoint, we would bring the Morse Group in to be able to provide that infrastructure that created the flexibility we needed. And ultimately, the design of the building was moving much faster than the design of the Lilly Life Sciences studio. And so we needed the ability to address that when needed. So that gives you a brief history of kind of how this evolved into the platform it is. This is a look at the L2S2 laboratory footprint in our Lilly Biotechnology Center today. We have 11,600 square feet that encompass the L2S2 platform. We also built the system with 3,500 square feet of additional expansion space. Again, thinking about the idea that we wanted this system to be able to evolve, we built that in from the very beginning. We also have several other spaces within the building. First floor for our NMR facility. We have a solvent storage room, and we'll talk more about the automated storage solvent delivery system that we put in place. We have an offline sample management center, machine shop, and then a server network facility. One of the things, again, thinking about relationship building, we also designated this space here as office space that's connected to the L2S2 platform for drug hunting teams, vendors who are working on site, the operational team that was actually operating L2S2, and other collaborators that may need surface hubs or spaces to sit while they're accessing the Lilly Life Sciences studio. So it's almost a separate operation inside of the larger Lilly Biotechnology Center. I'll also point out that we wanted this to be a showpiece. So this is actually the front entrance. The first thing you see when you walk into the Lilly Biotechnology Center, looking through the glass walls, is the Lilly Life Sciences Studio. Keeping with the idea that we were future-proofing this, we wanted this to be a very modular and collaborative system. So the track system, we could have put in a conveyor belt, we could have put in multiple robots that were connecting one another, but we actually went a very different direction. We put in a system called the MagnaMover Lite. It was a product by MagnaMotion, now owned by Rockwell. But essentially what it is much like a Hot Wheel track or a train track system. The configuration is actually adaptable to really whatever design configuration you want. It's made up of smaller points. There's switches, there's quarter meter rails, there's high precision rails, and so within about 30 minutes you can take a configuration and quickly change it into something else, and that's really the flexibility that we want. We've had a long history of being able to learn the goods and bads of automation systems, and one of the things that we found is the traditional automation, lab automation systems have become very hard to adapt for new things, and we wanted to take a different approach there. So ultimately what we have is a MagnaMotion system that acts as the backbone of the entire platform. We have the idea that we would be connecting integrated workstations, what we referred to as modules. We wanted not only to provide physical connectivity of samples going from one place to another, but we also wanted a conduit for all of the utilities, the house gases, solvents. We wanted those also to have a connectivity to the modules or the workstations. And so the track acts as our transport system. It also acts as our conduit for all the utilities that would be stubbed off to where the modules would connect. And so one of the things that you'll see here in this diagram is this is our module integration points. Every module integration point has a bypass lane, or essentially a drive-through lane. Every module has a standardized robotic arm with hotels, where essentially it acts as the liaison between getting things from the track onto the module. And the real benefit here was that we wanted the ability to have any integrated workstation built anywhere around the world and be able to, once we have protocols validated there, we wanted to be able to duplicate that system and have it go on to the Lilly Life Sciences Studio. Or if we could relocate a module from somewhere else or a workstation from somewhere else. And so a key to that is that we needed the workstations to have very few requirements in terms of connectivity. And so these, what we call the track to module robotic tables, give that ability to stage work that are going in and out of the modules, but also they have a robotic arm that can reach into the module system. This is a look at the underneath the track system, and this shows you an idea of what's at that track to module robotic table. So it's all quick connects. We have exhaust. The exhaust runs through the track system, and so when a module hooks in, you quickly connect the exhaust. Quick connects on all the house gases. We have a solvent waste system, as well as solvent delivery systems. All of the power, the UPSs, and the network cables are also fed through the track system, and they allow for quick connectivity and disconnectivity to the modules as we hook those up. So this is an early rendering of the L2S2 platform. And so one of the things you'll notice is you have the long MagnaMotion backbone, fork-shaped, and then you have the different modules that are connecting. One thing you'll see right away is that these are not identical modules. Every module is completely different and unique in terms of the hardware and the software that it has on it. It's also completely unique to the scientific application that's being run. I also want to point out that you have, by design, we have these, what I call bypassers, spurs, or drive-through lanes, and you'll notice the drawing shows several of these empty. We always wanted to have empty spots where we can quickly bring up the next set of technology, the next application, and quickly connect it to the system. Another thing I want to point out in this drawing that's very important is you can see a few people in the lab, and that was intentional, but what the people are looking at is a dashboard. there's dashboards on the wall, much like we see at the airport, where we see all of the jobs that are coming and going on the system. And so it was intended that the operators in the lab would have an operator dashboard of all the work that was happening at any given time. We wanted everything that was running on the L2S2 to be tracked, documented, and in a state where we knew where it was at any given time. Here's another look at that same system, again showing a better view of the spurs, or the drive-through lanes, and then just to give you an idea of some of the different instrumentation that was on the modules. Now this is a video where we're actually showing you the automated connection of the modules, and one of the things I'll point out here is look at the drive-through lanes. Also the camera system. We worked with the Morse group on the camera system. We brought in the same camera technology that's used in casinos for locking onto people and then tracking them, tracking their movements across the casino. We wanted the ability to lock onto a sample and follow that around the system every step of the way as we went through the modules. So this highlights the vehicles that are running. It's a magnetic levitated system. You can also see the exhaust that's ports that are going, so everything on the track is exhausted for safety reasons. I want to take this moment to then touch a little bit on batch automation versus continuous automation. Most of the lab automation knows of automation, and when we think of automation and lab and science, we think mainly of batch automation. Batch automation essentially is built on what is most efficient for the operator. So if I'm going to have a human come over and interact with a system, I want to be able to load up as much work on that system as possible and then go walk away and do something else and have the automation working. We can equate it kind of to the ferry analogy. You have certain time points where people can get there, you load everyone onto the ferry, it leaves, and now everyone else is kind of waiting. The difference with L2S2 is that it's built on continuous automation. So continuous automation is an analogy similar to a toll bridge, where cars can come and go as they please, but then there's checkpoints or tolls essentially that allow for some sort of batching and time release. This system was built with the continuous automation in mind. So we want every sample that's running through the system to be able to process and then go on to its next step. And you can imagine when you think of automation in this way, the details you have to think about in terms of, do I want every module to have my own supply of consumables, or do I want to have central consumables and building blocks and reagents that when a module needs it, it can flag and we have that sent. You also had to think about things like, if we're not going to do batching, do we really need huge, liquid handlers and deck space and large incubators and carousels? So it's a little bit different thinking, but I just want to point that out, that Lilly Life Sciences Studio is really built with continuous automation in mind. And so one of the things that we did to make that possible is that every module had to be autonomous. It had to be independent in its functionality. We can have multiple processes running on a single module, but the system as a whole will have multiple modules running in parallel and be running totally different science. And so this is a look at one of the early renderings of our sample management and reformatting suite. And one of the things you'll notice is, there's an automated storage unit, there's an automated weigh station, there's a acoustic low volume reformatting module, and a traditional liquid handling formatting module that's being shown here. Each module has scheduler-specific software, and so one of the things we wanted to do with this project is we didn't want to make this a custom automation project. We wanted to use as much commercially available hardware and software as possible on the market and make this really a custom integration project. And so each one of the modules has its local scheduling software, and this may be Venus Dynamic Scheduler, Instinct S, Green Button Go, WinLizzy, AutoSuite, Ignition, but we didn't want to reinvent the wheel there. We want to leverage whatever the collaborator or integration company was using, and then monitor once things get into a module. Essentially, that module became the master. and so the module was controlling everything that was in the system, and then sending information back and forth to a higher level scheduling software. And so this is just kind of highlighting that there's autonomous module workflows that are happening. Each one of the modules can have multiple processes running in parallel, and all of those are being tracked, and then they're being fed up to a higher level software. That higher level software is the vision that Lilly had for that was called the master scheduling software. Essentially, this acts as the air traffic control tower of the system. We needed a scheduling software that would handle order management, being able to receive orders, parse parameters, and then feed those parameters to the appropriate modules on the system. So one of the things the master scheduler has to be able to do is it has to be able to understand the capabilities that the platform has. And when an order comes through, it needs to be able to take that order that could be an entire drug discovery process and break that into meaningful parameters and components that it can then interface with the modules. And so in addition to module integration and being able to do bidirectional communication, the system then had to coordinate the platform scheduling of everything that was happening on the system. It had to be able to do air handling. So on a system of this size, you can only imagine the types of airs that would be coming up and the ability to handle those in real time and divert and dynamically change. So it needed to be truly dynamic. In addition, it needed to be able to do all of the data processing. So it had to be able to manage data flows. And then a key to having a system that is fully automated is you have to build the intelligence into the system where when things happen, it can automate the decision making. And so we built a platform that continues to get better over time. The more and more runs that occur, the more data we have to feed the algorithms and make those decisions. And then we needed something that provided a platform dashboard. And of course, all of this was the function of the master scheduling, but then it had to interface with the outside world. So we talked about we wanted to open this up to a global network, and so that was through our cloud-based user experience software highlighted there. We also have internal LIMS databases, ELNs, local servers. We also knew that there'll be new LIMS systems and other researchers with different LIMS systems, so it had to be a bit genericized so that it could fit with other people's needs. And ultimately, as we assessed the market and put out an extensive RFP, we partnered with Biosero to develop the master scheduler for us. And so I wanted to highlight just briefly what they came up with to build this concept. And so they started with GBG as the backbone. They came up with GBG Order Manager or Order Broker, which essentially takes in orders and then dynamically creates the workflow, and then it can parse parameters to send that out to the different modules. Biosero controlled the track system through the Transportation Manager. And that gave the ability to know everything that was running on the track at any given time. It's a bit like an Uber system. So when a module needs to send something out to the track, it flags, and a vehicle pulls in, it can hand it off, and then it goes on to its destination, drops the passenger off, and now it's available again. They were able to handle those track-to-module robots through what was called the multi-robot service, and that's a plug-in. And then the way the modules were actually integrated was the ability of creating what was called a module agent. It's essentially a driver for a workstation, and that created the ability to translate information coming in from the master scheduler to a language that each module could understand. Here's a look at some of the schedule interfaces, the ability to take a module on and offline. I mean, one of the things that we have to be able to do on this system is do PMs and downtime. So the ability to quickly take a module offline while the rest of the platform continues running. And so there's quick buttons for that. There's also the ability to dive into everything that's on a track on a particular vehicle and see the contents there. And ultimately, what happened with this and with many other things that Todd will talk about later is this idea that Lilly did not want to build a one-off custom system that would become a dinosaur after day one. We wanted to partner with vendors and collaborators in the market that saw the benefits of actually taking this further and getting feedback from the rest of the market to productize this. And so Biosera was able to take this concept and productize it. They now have it as components of their product, which are being showcased here at SLES. In fact, the GBG dashboard is one of the 2020 best new products as well. And so, highlight that and the work that was done there. This is actually a video showing the master scheduler in motion. And what you're seeing here is exactly that Uber system where it's an order's been placed. This is actually an acoustic dispensing method. So we have several source plates that are going to create a destination plate. And so the system right now is actually getting those the source plates. One of the things you'll notice is that there's automatic traffic control of the system, so it knows where other vehicles, it knows how close it's getting to other vehicles. This is a look at the track-to-module robot transport station. One of the keys of being able to keep things properly scheduled and have the ability to dynamically change is having this idea of this buffering station. We wanted to be able to have a place outside each module where we could queue up work. So you may have a reaction preparation that has to occur that needs 7 different building blocks and reagents that are coming in different formats. We don't want to tie up a module while all those things are being gathered. So this robot transport station, the buffering area, gives the ability to bring all the components that are needed for an order together and then start queuing up the module once they're there. So this is now showing loading into the module. In this case, this is an Echo Access system that's been integrated to the L2S2 platform, and it's now taking in the different source and destination plates and running that. Many of you are familiar with the Echo Access, so we'll continue along. But on the back side, after this is done, all the work goes back out to the track system and on to the next thing. In addition to the hardware and the automation scheduling software, the interactive experience of L2S2 was one of the highest critical components of the system. Much like we use Amazon today, and we use other apps. We either love an app or we dislike an app within a few minutes of using it. And it's all about that interactive experience. So we wanted a system that allowed people from all over the world, easy access, log in, and then at their fingertips have the ability to tailor that experience for themselves. And with this, we wanted to give them the ability to see what building blocks, reagents are available for synthetic reactions. We wanted people to know the time and the cost associated to those reactions. And then once that order is submitted, we wanted the ability for people to experience the automation process as if they were doing it themselves. And so we wanted real-time access to data. We wanted them to be able to log in and see the picture of what their reaction looks like, of the color change. We wanted the ability for people to see the video footage and track their system along the way, and all of that is part of that user experience. And with that in mind, we wanted this system to be opened up broadly. Lilly has some history of having automation systems with remote access, but it was always very a lot of stipulations. You had to have a Lilly device and that sort of thing. We wanted this to be different, and so we wanted a cloud-based system that was accessible not only by just Lilly but by other researchers. And with this, we partnered with Stratios to bring this capability onto the platform. Stratios has a long history and experience running one of the first cloud-based robotic labs. In addition to tapping into their experience on the cloud-based infrastructure, we also brought in Stratios to operate the lab. It was key to realizing this vision that if Lilly is going to open this up to other scientific and pharma and biotech organizations, it can't be Lilly that's running the lab. And so we brought in a third party that would actually operate the lab and run it as a business. And with that, Lilly has provided Stratios the ability the freedom to operate. And with that, explore handling portions of the master scheduling software, hiring, staffing appropriately, and performing the user experience and that sort of thing. And we'll talk more about that. I quickly want to go through just the project timelines. We broke this into 5 phases, and you'll notice it started July of 2015 through Q4 of 2019. We had a project planning phase for a year and a half. We moved forward with the initial track-based infrastructure and sample management and reformatting center. We then added synthetic chemistry capabilities. We then went into biological testing functionality, and really 2019 was geared around validating the science and continuing to deliver out on that full user experience. This is a look at how the system came together. So this is phase one. We have the track system, sample management and reformatting. We then added synthetic analytical and purification capabilities, some additional green boxes, and then our biological testing capabilities. And as mentioned, this system is constantly evolving, so we have development modules. One of the things I want to point out very quickly is the green boxes highlight the only 3 modules on the platform that were intended for regular operator intervention. You can imagine that if an operator had to go around to 16 or 20 different modules and take trash out and make sure what tips are loaded and solvents and wash buffers are there, that would kind of defeat the whole purpose of having it fully automated. So we made the decision that we had 3 points on the system that were going to be geared for operator intervention, and everything else would operate in a fully automated manner. These points also gave us the ability to check in things to the system and also check out. So at any given time, we knew everything that was on the platform, and we know everything that's coming off of the platform to create that positive sample tracking. This is just a look at the breakdown. So we have really 4 suites. We have a sample management reformatting suite in blue. We have a synthesis analytical and purification suite in red, and then we have a biological testing. We have development modules, and then we have what we call our user interface suite, and that's the green modules. The construction timeline, we did the building, finished in December of 2016. By December of 2017, we had the lab space done. By the end of 2018, we had many of the modules and systems in place. And then by the end of 2019, we have the entire originally planned system built. Just a few facts and figures. We have nearly 12,000 square feet of laboratory space that's fully automated platforms. We have over 500 feet of magnetic levitated track system. We have over 28 collaborative robotic arms, over 100 automated instruments. If we talk about the automated devices that are on each instrument, we have several hundred that are in the lab. We have over 30 instrument controllers. We have an automated consumable capacity to hold up to 4 days of consumables for up to 4 days of operational productivity without human intervention, and that's at peak capacity. We have the ability to hold over 5 million compounds at any given time. And one of the nice things in using the Verso store is we can actually have those building blocks, reagents, compounds in a variety of different formats. As of right now, we have 16 connected, fully automated scientific modules that range from synthetic capabilities to sample management all the way to biological and biophysical testing. Everything on the platform is being captured, so data is key. We're capturing as much data as possible to continue to refine the algorithms for machine learning and artificial intelligence, and that's at the process level. We're also capturing all of the scientific data as well for that reason. I want to give a quick thanks to all of our automation integrators that have made this possible. You'll be hearing more about them as the talk goes on, but these companies were all key to allowing us to turn our vision into a reality, and we want to thank each one of them for making that possible. This is a map that actually highlights the modules that each one of these vendors worked on, and I must say, other than a conference like this at SLAS, I'm not sure that there's ever been a single lab or a single platform where we've had all of these different vendors and integrators come together with a shared goal and really making this possible. I think that's been unprecedented and a remarkable story in and of itself. Last but not least, I wanted to highlight our operational partner Stratios and for the partnership we've had with them, and really them allowing us to make it possible to say we're going live February 2020. And so I'd like to now take this opportunity to to give you the first look at the Lilly Life Sciences studio. Hope you enjoy. Lilly has a long history of being a pioneer in automated synthesis. In 2015, we thought about the need for doing something more. Could we go where no one has really gone before in automating the entire drug discovery process?
My role was to lead a very creative team of scientists and engineers to create an unprecedented lab, but still deliver on time and on budget. It took us 3 years to build the system, including a first year for construction of the lab space.
One of the initial drawings that I did of what the lab may look like, where we drew out the concept of having fully automated modules being overseen by a higher level automation scheduling software. It really acts like the air traffic control tower at an airport.
We partner with multiple companies and we asked each one of them to bring their A game. And it's actually been amazing to see the system being built in several sites and locations across the US and Europe, and then being shipped here and assembled all together in the lab space.
So now we have an entire ecosystem that is physically connected and virtually connected to get answers sooner.
A process that through more traditional means can take up to 1 or 2 weeks. In this laboratory, we can do it in 2 to 3 days.
When we were planning the Lilly Life Science studio, we were really focused on what we called the 4 R's. So we wanted something that was reproducible. There's really no better way of doing that than with automation. We wanted something that was responsible, and we achieved that by reducing our footprint of the amount of reagents and materials we're using. And so we're being a responsible steward to the environment. We wanted something that was relationship building. We wanted it to be a collaboration hub.
The thought there is that it will be accessible not only by Lilly scientists, also by collaborators in biotech and academia outside of the Lilly walls.
We wanted something that was rewarding. We built Lilly Life Sciences Studio for the next generation of researchers out there who want to make a difference in the world, a system that can continue to evolve as technology evolves and as research evolves, so that we can ultimately make life-saving and life-changing medicines for patients around the world.
The Lilly Life Sciences Studio literally reinvents the paradigm of drug discovery. It's a first-of-its-kind research ecosystem. It's a resource available to scientists to empower hypotheses in a way that's never been delivered before.
It's just really exciting because nothing like this has ever happened before. A scientist can design compounds and have them synthesized and then test them in biological assays, all in an integrated fashion.
The studio comprises research functions that have historically resided in individual departments or organizations. So each of those functions may operate very effectively, but as a process whole, it's a rather ineffective process. And so the Lilly Life Sciences Studio, as an ecosystem, brings together these components from the design of a molecule to test a hypothesis, to the synthesis, all the way through to the purification, plating, and ultimately the biological analysis of a sample, and provides real-time information to the scientist versus what's historically can be a multi-week process.
In a typical lab, you would have operators that would move your samples between the different islands of automation. With the Life Sciences Studio, we now have a MagnaMotion track that ties together all of these different areas.
This is also a paradigm shift where instead of a robot moving to a sample, you now have the sample moving to the robot.
Even though all of the modules are really considered islands of automation, these are all tied together through the master scheduler so they can all communicate with one another.
This allows us to have a system that is scalable, flexible, and maintainable.
It gives us the ability that as new cutting-edge techniques and science come out, we're able to quickly add those onto the system and open them up not just to one researcher, but to a broad research collaboration.
The submitter can be anywhere in the world and present work to the studio for work to be done. Once the order is placed, a lab technician in San Diego will take the compounds and place them in the input-output module. Once that happens, the master scheduler will request those samples to be moved to a new module for processing. We have a very extensive camera system that allows a remote user to track their sample as it goes through the system. The requester can then get his data through a web portal and find out exactly what was made on the local system.
So for Lilly scientists, this laboratory will move them to the next level of productivity and insight. For example, when scientists are at home with their families, the lab will still be relentlessly executing their experiments.
The most exciting thing about the Lilly Life Science Studio is that it's going to be addressing some of our needs to reduce cycle times and go faster on projects to make go, no-go decisions. And ultimately, it's that innovation that's going to be able to bring the right drug to the right patient in a timely fashion.
We want the outward-facing experience for the lab to not just be for a medicinal chemist. We want it to be approachable by people with different disciplines than a chemist, biologists, physicists. One way we do that is that the software that a user would interact with will allow them to design molecules using a drawing tool, but then it can take that molecule and retrosynthetically break it down into the building blocks that would be required to make it, as well as determining what that recipe is. Once you have that put together, you submit, and then all that complexity of actually doing the synthesis is all taken care of by the software.
The system can also be used where a chemist wants to see what can be made on the platform, and that's where the system could recommend compounds based on availability of starting materials, the type of chemistry that the system can run well. So there's different ways that the submitter can actually engage the platform.
Chemists spend a lot of time trying to find compounds. It's not a defined science. It's kind of an art. The software will determine what building blocks are in inventory right now, what has to be purchased. It can give you what the price was going to be, what the time period is, because certain things take a while to get. So the ability to take all that away from the chemist and have it all automatically generate, that's another big, huge advantage for the system.
One of the main differences in how scientists use the system versus the previous platforms is that the deliverable before was compound in a bottle, where the deliverable in this platform is the data associated with that compound, which is a key difference and really allows for a wider user base to access this platform.
The sample management and reformatting suite currently has 5 modules. It consists of 2 automated stores, an input-output module, and then a weigh module, and a module for reformatting samples. So the Verso stores actually allow us to store many different container types. What is unique about them is we can now pick these different type of containers to build orders, and we can actually pick millions of samples to run on this system. So the input-output module allows the operator to have one touch point to be able to put samples on the system and take samples off of the system. This is very important because we really want to do positive sample tracking of every sample that's moving on the system at any time. In the high-volume reformatting module, we have the ability to reformat samples between the different vial types, tubes, and microplates that we will use in the different assays. The low-volume reformatting module allows us to build assay-ready plates to be used in our biological testing assays.
One of the key technologies with this platform is the reagent encapsulation, where this system can dispense solids, oils, viscous liquids, and deliver them into encapsulated reagent containers that, just like a coffee pod that is just a single serving, the right amount, the right quality, the right strength, the waste is minimal. That's how we've approached storing and delivering reagents to reactions using just the right amount of material. This just-in-time reagent paradigm that we're implementing on this platform brings a level of standardization and quality to the sample management storage process.
The synthesis suite of the Lilly Life Sciences Studio leverages a decade worth of experience in automated synthesis by Lilly to create an entirely new way to do molecule design, synthesis, and purification. It includes the synthesis and analytical purification module number 1, where reagents are combined into a reaction vial, and then that reaction vial can be staged to either run immediately or run at some future time that the scientist might prefer.
When we looked at the synthesis suite of the Lilly Life Sciences Studio, we looked at new or innovative solutions to create a fully automated synthetic chemistry workflow. The pinning technology is a very new concept, and we were the first company to embark on this type of fully automated reaction preparation. Unlike previous platforms where we had to do each step discreetly and independently, now this technology and the way that we've designed this platform, multi-step chemistry in a single vial, is a reality. This de-siloing of different scientific functions now allows a compound designer such as myself to be able to design a molecule, and then that biological data is captured all on one platform without me having to make multiple requests. What would have taken a few weeks to a month to go through that full design cycle is now down to days to hours.
At the time this amazing adventure started, I was asked if I wanted to join the team, and I said for sure yes. I had no doubt. I wanted to be part of this team. For the very first time, we will be moving from crude reaction to pure isolated material in a straightforward and fully automated manner. What we want to provide our chemists, our scientists, is a very, very efficient way to get data that will allow them to do the right decision very quickly, so they can really accelerate the progress of their research projects. This is what is really important here.
We generate roughly 15 to 20% annually of the entire Lilly compound collection that goes to biological screening. We then took the next step and began designing an ecosystem that included the automated synthesis component, but additional components as well, including purification, analytical, and then ultimately biological testing.
The experiments that I run are run on benchtop automation equipment, and a lot of these routine cell-based assays and biochemical assays we can put on the Lilly Life Sciences Studio, with the convenience of coming in the next day to work and having an email with all the data. Science is always evolving and advancing, and one of the great things about the Lilly Life Sciences Studio is that it's designed to expand and evolve with it. So as new scientific technologies are being discovered and developed, the Lilly Life Sciences Studio is always relevant and current.
Once a compound is produced, this lab has the ability to then do a second round that will be the biological assay and gather the biological data. So not only can you make the compound, but then you can make a decision that you're going to look for activity through this range of biological processes. And then you're even one step further in making a decision on whether this is something that's valuable or not.
With the Lilly Life Sciences Studio, we wanted to make the experience for the submitter and the customer as close to how they would be working in the lab themselves. So the combination of the analytical data with an image helps to inform the scientist in real time the state of their reaction.
Key component of this lab is the ability to track the data from the very beginning of the order all the way through to the end. We know all the conditions that it ran through. We know the different reagents that were used. We know the building blocks. We know where they came from. We know how long they'd been stored. So we have data on every possible component of the process. And if something didn't fail, or if it came out really good, we can reproduce exactly what happened.
The standardization and the quality of data that we're collecting on this platform makes the ability for the system itself to make decisions or recommendations. Say, this reaction only went 10% complete, but it went 10% complete because the wrong solvent was used, or we didn't heat it to a high enough temperature. As a chemist, I can look at that data and make that decision. But if I was not a synthetic organic chemist, I would look at that data and not know what to do with it. Now we have a system that is able to analyze that data and then make a recommendation on that next experiment to be run and be able to launch that experiment on behalf of that submitter. So the submitter doesn't even have to be involved in that resubmission.
We also have a vendor that specializes in data analysis, and they produced a completely novel package for us that runs on one of the modules, is coordinated through the master scheduler, and it collects data through each step of the process, uses it to make decisions, but also gives you this complete data package at the end that submitter can then use to decide to go forward with that compound, to modify the compound, or to abandon that effort. Also, not only can you make the compound, but then you can make a decision that you're going to look for activity through this range of biological processes. It will take that compound, resuspend it, run those assays, get that data, and then you're even one step further in making a decision on whether this is something that's valuable or not. It is doing the work of an entire medicinal chemistry lab as well as a quantitative biology group. When I first saw what they were trying to do, I kind of thought they were crazy.
The Lilly Life Sciences Studio represents a paradigm that has never been done before.
There's never been anything like this done ever in the world. Nothing like this exists.
Come and join us in this stream. It's a story of trust, engagement, commitment. This is reality.
So I want to thank Ryan for the introduction to really describe what the L2S2 is. The video reveal. First off, I want to thank our corporate communications group and the members of the team that spent the time to make this video and to be able to, the opportunity to share it with everybody today. And as Ryan alluded to, the video will be available on Lilly's website as well as Lilly's YouTube channel, hopefully, I think, after this talk. My portion of the talk with the time we have left is going to be, and again, I also want to make sure to thank Hamilton for the food and everyone's attendance today. We're just honored to be able to present on this endeavor and the team that we got to work with to build the system. So Ryan described the what of the L2S2, and my goal in the remaining time left is to highlight really the why we built L2S2, and through a scientific and technical overview, some of which you saw in the video, I want to describe really the how of the L2S2. And then Ryan's going to get back up on stage real quick, highlight the opportunity for the system to expand over time, and then we're going to transition to a roundtable discussion with my Lilly colleagues that were on the original scientific and design team on the automation and scientific front. So there's an evolution of how R&D is performed in the life sciences industry today. Kind of going back a few years, and speaking from Lilly, Lilly's the only company I've ever worked for. Most of the work that we did early on in my career was the scientists and the science itself was done within the walls of Lilly, internally owned, and mostly through manual processes. We evolved to running outsourced R&D using partners that had a specialty in that particular field of science, biological assays, chemistry, outsourcing, et cetera. Lilly then moved to an in-source model to bring some of those partners that we have identified to be on-site, co-located with Lilly employees to allow for a quicker cycle and getting that data into the hands of the scientists. And all through that, there was initiatives going on at Lilly around automation. But what we like to call, and Ryan referred to it, is these islands of automation was really the key strategy at Lilly, where even though we had a major system that would run a good bit of automation, we were still dependent on humans to either walk samples from lab to lab, or individual steps within that automation process would require a human. And kind of the holy grail, as in the literature, especially the last year or two, there's been a number of review articles and opinion pieces around the closed-loop pharmaceutical design. And one of the constants in all of these literature reports, as well as what we've hoped to achieve with this this L2S2 concept, is achieving this closed-loop pharmaceutical design where everything in that cycle is within a single ecosystem and using and leveraging as much automation as possible to achieve that closed loop. And you'll be seeing this closed loop concept throughout the rest of my talk. Now, the history of automated synthesis can be its own talk. I'm not going to go through the long history other than to say that we've been doing automated synthesis now for over 50 years, and really what I want to highlight is the items in red, where we've evolved from peptide synthesis systems to CombiChem that was run 20 years ago. And for some of you in the audience, this is the last time that you've seen automated synthesis run was in the CombiChem days. I can assure you we're not doing CombiChem anymore in drug discovery. It's evolved, and really over the last 10 to 15 years, as Jim Beck alluded to in the video, we've got we had a system in Indianapolis that was able to do parallel medicinal chemistry very effectively, and that has evolved now to what we consider hypothesis-driven chemistry. Just because we can make a compound doesn't mean we want to make that compound. We have to have a hypothesis, and we deal with quality, not volume anymore. And with that, there's a number of software, equipment, and chemistry evolution that has taken place over the last 50 years. So really the last 10 years has focused on the automated synthesis and purification lab, which ran in production from a little over a decade, from 2008, and it was just decommissioned late last year. That was based in Indianapolis, and the acronym ASPL stands for Automated Synthesis and Purification Lab. We had the ASL component and the APL component, the Synthesis and Purification Lab, and this is these labs were right next door to each other at our corporate center, and one did synthesis, one did purification, but they had different systems, different operators, different processes, but there was a high level of connectivity, especially with the data. And the approachability of the scientists being able to engage one or both of these platforms to execute the science that they needed to run in an automated way to run. And the other thing, one of the things I want to highlight is probably the very first question I get as a chemist that's worked in automation now for close to 17, 18 years is, what kind of chemistry can you run on automation? And I like to point people to two really key papers. One on the left-hand side is a paper from Novartis that was published in 2016 that highlighted that did a really nice analysis of the last 40 years of chemistry that was run in the patents and literature, and really kind of define what the medicinal chemists and synthetic toolbox is, what were the most popular chemistries, and the distribution of those chemistries. Also in the same year, one of my colleagues, Christos Nicolaou, wrote a paper around the proximal Lilly collection, which has been well documented and written. But that analysis covered the first 5 years of the ASL, 37,000 reactions that were performed over the first 5 years. And when you look at this in a little more detail, the chemistry types as well as the distribution almost match identically. So, long story short, any chemistry that a chemist is running at their hood, the most popular chemistries, we've been able to demonstrate that we can run those in an automated way. Now, obviously, there's going to be exceptions with specific reagents, or there may be a specific amide coupling or acylation that is problematic on the automation that really deserves to be in the hands of a chemist at a hood. But overall, most chemistry that we can run at the hood, we've been able to demonstrate and prove that we can run on the automation. So I want to just highlight an example that occurred in 2015, which, when Ryan described the calendar, was right about the time that we started really thinking about the next generation automated synthesis and automated synthesis platform. And this was an example where I was approached by a computational chemist within Lilly. He was based in San Diego at the time. I was in Indianapolis in 2015, and he had this hit that he wanted to expand on. And this chemist had very, very little synthetic organic chemistry experience. So he came to me and said, Todd, I know you're an expert in the ASL. What kind of compounds can we rapidly access for me to expand this area of the protein? And immediately there was a commercially available halide and over close to 1,200 different amines that we could stitch in to form this NH-linked compound series. And through the compound design process, him and I were able to prioritize the top 50 compounds in this set. And again, this hopefully will be a good example of that next step down from the closed loop, and to really highlight that we were still looking at a siloed automation process specific to the application. The molecular design step was still highly manual. We were kind of going back and forth, and there was a lot of manual work that was being performed with that. The computational chemist was dependent on an ASL expert to access this automated synthesis resource effectively. And then the chemistry process itself was semi-automated. But you can kind of start seeing that we've got a loop going here, but there's certain steps that are still manual and still suboptimal that we saw as opportunities to improve the process. Now, to kind of go a little bit deeper with this example and really highlight some of the things that were well optimized and things that had room for improvement. The good news was we were able to rapidly expand 3 different vectors of this SAR, kind of going out to the right, out toward us, and then kind of out and back, using 3 different linkers, a nitrogen linker, an amide linker, and what we called the reverse amide linker, where the carbonyl was closer to the core. The good news was all 3 of these conditions were from my institutional knowledge, I already had these conditions well optimized from a previous chemistry campaign. So I didn't need to go and find the best set of Buchwald conditions, amide coupling conditions, et cetera. The success rate of the reactions, we had about a 77% success rate. 122 out of 158 reactions gave me enough product to test. And again, that was the name of the game in this stage of the process, where we just needed enough compound to get data on to make a decision. And in fact, the computational chemist was thrilled with the success rate. In fact, we didn't need to go back and remake any of the ones that failed. Now, talking about some of the manual steps and some of the processes that had room for opportunity, was that with this being a shared resource, the ASL was a shared resource. I believe there were 30, 40 chemists using the system daily. All 3 chemistries required the microwave reactor to execute this chemistry. And one of the things with the ASL is that it had no forward-looking scheduler. It was first in, first out, and there was no scheduling component of the system. So what I had to do, one, to be a good steward of the system and not have my colleagues get angry at me for submitting 158 reactions in a single shot, is that I had to manually stage these reactions across 20 business days, and that sweet spot was around 6 to 10 reactions a day. So from a scheduling point of view, I had to own that schedule, and because I had knowledge of what was a good steward of that resource. Also, from the reaction scale, I wanted to make sure that I ran these reactions at a high enough scale to tolerate a wide variety of yields, but not at the same time balance the optimum scale that the ASL and, more importantly, the purification lab could handle in a quick way. We didn't want to have multiple fractions, pooling, long dry down times, et cetera. So I had to come up with that optimum scale myself to get this campaign to be as successful as possible. But bottom line is the bulk of the work for me was opening and closing electronic notebook documents, building the automation files that were used to submit to this platform, and then a manual review of the data. But the good news is neither me or the computational chemist needed to step foot in the lab. So I think from a efficiency standpoint, he was able to go off to work on other things, and this was a secondary project of mine at the time where I was still able to work on another project that was my primary project, and so forth. So before we get into the L2S2, the last slide I want to show is this ended up being one of the main use cases that we used to really interrogate the existing processes that we had at Lilly and look at the opportunities to improve this semi-automated process. And there were really three areas where there were opportunities to either reduce or eliminate by an operator, the number of manual tasks that were performed by the operator. I'm not going to go through all 10, but one of the first ones is that with this system, we had to manually set up all of our reactions manually, since we had one functionality that we did not have on this platform that you saw in the video was the ability to use solid handling and some of those technologies that I'll highlight in detail here down at the later in the talk. The system only could handle liquids and stock solutions, so that's a key manual step. The very first step was dependent on a human to be in the lab to launch this work. And then there were also a number of manual decisions that needed to be made by the submitter. Again, being a good steward of the system, we had this park-pause opportunity on the ASL where we could review the data before sending it to the purification lab. So if there was no compound, I wouldn't waste that submission to go to the purification lab, tie up their time, tie up the resource when it could be used for other chemistry that was doing well. And then just the manual review at the end, reviewing the structure, registering it, sending it to sample management for plating. So just to show that, even though we were running things semi-automated, there was a wealth of opportunity to improve the process, and our team used this as one of our use cases to improve the process. And that really gets into what I said in the video and what Ryan alluded to, is that the output of the previous paradigm was compound in a bottle. The molecule was our end product, where the L2S2 and where we wanted to go was that we wanted to go from idea to of course, data. Because at the end of the day, really, the scientists are more interested in the data than the compound itself. So Ryan showed this slide earlier around the idea to data. I'm not going to go into this in great detail, other than we feel that now this paradigm is really approaching this closed-loop drug design paradigm that we want to achieve. So to show the slide again that Ryan showed earlier with all the vendors and the layout of the lab, that again we had 3 touch points with the green modules. We had our sample management, synthesis, and testing suites, all in different colors here. Usually with the technical and scientific overview, when we give a tour of the L2S2, most of us usually enter in through this door right by the lobby that Ryan showed, and we usually walk around counterclockwise, looking at each one of the modules and giving a tour and an explanation of what each module does in terms of its scientific and technical capabilities. When Ryan and I were putting this talk together, we looked at this and said, really, is this the best way to showcase this? Because the way that the lab is laid out is not necessarily the same order in which the overall process goes through from beginning to end. So we thought about this for a while and thought, it would be a good idea to show this story called Follow the Molecule, which is in the same line which aligns really nicely with this design, make, test, and analyze paradigm that we wanted to achieve, but intermingled in with that science and technical work that the lab does is a number of operational components around feeding the system, storing reagents on the system that you use for the synthesis part, as well as the products that you make that go in for testing. And it's really worth highlighting here that you can have the most awesome hardware in the world, best software to run this lab, but if you're not effectively able to fuel it and keep the system running from an operational point of view, this capital investment that Lilly invested in, we weren't able to maximize the use of it without a robust operational system. So the first step in the process is compound design. And I'm a guy that likes to use a lot of analogies, so hopefully I don't use too many, but one of the ones that I like to use as a chemist is and a simple thing is that with the compound design, there's really 3 steps that you look at. You design the compound, you design the route, and with that route, you choose your reagents to execute that route. And I'm going to use a really simple example here that I want to make this compound. And what do you need to make a compound? It's just like cooking, right? You need 2 things. You need a recipe and you need a shopping list. And with that, the recipe, ideally, it would be really nice to have that recipe that has some type of automation feasibility associated with it. If we are able to choose a recipe that already has automation protocols with it, and this is where our retrosynthesis and informatics tool that was built in-house called ChemoPrint comes into play. And in this case, the chemistry here is really straightforward. It's a Suzuki coupling, carbon-carbon bond formation, and ChemoPrint, this software that we've developed in-house, of course, predicted 12 Suzuki reaction conditions that could be used to stitch this compound together. And what's nice is it ranks them by chemical feasibility and where the compound was synthesized, the recipe that was used to synthesize a similar type of molecule. So in this case, there were four out of the 12 had a high degree of automation enablement, and then it also looks at reagent availability and cost. And this gets into one of the things that you're going to see throughout the talk around the standardization of reactions, that one of the key things that from a foundational level is we needed to standardize our chemistry at specific scales, because with this ChemoPrint tool, as well as the instrumentation that we put into our chemistry and testing suite, we wanted to ensure that we were making chemistry just enough to make that material to test, get data on it, and then have an automated recipe to iterate if need be. And it's also worth mentioning that our colleagues, Terry being one of them here, has a manuscript that's just been accepted that's going to describe ChemoPrint, so keep a lookout for this article coming out. So in addition to compound design, you may have the best recipe in the world, but if you can't find the ingredients to perform that recipe, you're going to have to figure out another way to make that recipe or find another source of your starting materials. So the reagent availability cost analysis combined with the reaction feasibility, you can quickly see a lot of these reagents were very inexpensive to obtain, or they were available in-house, where some of them were either commercially unavailable or prohibitively expensive. So between the cost and the route and the recipe, we were able to prioritize and look at a couple reactions that kind of floated to the top as our favorite reactions. So the first one used one of the partners was a bromide and the boronic acid. And the data that we showed, the first reaction that we ran, we saw absolutely zero product. So not fantastic, but as I alluded to in the video, this LC data is really only valuable if you also have an image. And me being a chemist, when we were validating these ChemoPrint predictions, the first thing I noticed is that the image of the starting, this was before heating, this was after heating, it looked pretty similar. And it turns out this boronic acid is highly insoluble, and even at elevated temperatures never went into solution, and thus the reaction didn't go. So the second attempt was just simply swapping out one of our 2 starting materials to boronic ester. And as you can see, before heating looked very similar, but after heating, well, we had a color change with that yellow solid was gone. And lo and behold, we had a brand new peak. That peak was indeed our product, and after purification, we got a 65% yield. So just in 2 attempts, we were able to get the product of interest in enough material to be able to move forward. Kind of stepping back to that example that I showed with the computational chemist, if he would have had a tool like this where he could have just drew that molecule, he could have potentially gotten one of these sets of conditions with the materials that would have been readily available and been able to submit work to the automation platform, whether the ASL or L2S2, without me being involved. So the dependency on having a synthetic chemist would not be critical in this case. And one analogy I've used over and over again, but I think it's really fits nicely here, is that chemoprint, in my mind, just hand me a dart and blindfold me, spin me around, and then tell me that the dart board is on one of the 4 walls. What chemoprint does is steers me to the wall that the dart board is on. So I have a better chance of finding that set of conditions. This is where chemoprint, and this chemoprint evolves, and as our confidence in the procedures that chemoprint provides, we feel that this is going to really be an important step in ensuring that we have a good starting point in compound design. So the next step is feeding the lab, and obviously the standardization comes in. Ryan alluded to this. All of our labware is standardized, and it's also very amenable to existing automation. All of the vials are used either in existing platforms. We have 2 vials that are our reagent vials. We have 3 reaction vials that are our Biotage microwave vials that are used across all of our reactors. Our collection vial for prep purification is in a taller vial that can hold 33 mils. Our LC mass spec and NMR vial are also barcoded. And then our SPE cartridges to do pre-purification on our platform. We have a 20 mil and a 35 mil cartridge that successfully our integration partner that built the workup system was able to successfully integrate both of these size cartridges in a fully automated process, which was really something that we were proud of. But what was nice is that this, now having this size, and again, this gets back to the 3 scales, we were able to fit all 3 scales within this set of standard labware and not have a ton of labware to deal with. So we really wanted to simplify this. The way we fed the labware, the way we feed the labware to the platform is in 3 different modules that Ryan showed on the map in green. The first one is a consumable store that was built by the Morse Group that has room for 384 SBS racks. Ryan alluded to earlier that we wanted to have this lab be able to run for 4 days unattended. This matches in with Memorial Day weekend, 4th of July holiday, and one of the limitations that we had on our previous platform are we only had 24 input positions. So once those 24 input positions were brought into the automation, you were dependent on an operator to feed it 24 more things for the automation to continue. Having a large capacity at the front end eliminates that issue. Now, the other input-output modules, we have 2 of them on the platform. One is in the sample management area that mainly deals with our shorter, smaller labware plates generally, and also our vials that would handle both building blocks as well as reagents and compounds for that are already going on the platform that don't need synthesized but need stored for testing. And then our other module over in the chemistry portion of the lab is our input-output module for synthesis, analytical, and purification. And instead of using hotels, Biosera was the integrator or the designer of this platform. They integrated this really nice stadium seating to be able to have 4 data trackable bays, an input bay for vials that, unlike the consumable store, which were for clean vials, if we had a reagent that was already in there or even a reaction that was run offline and the scientists wanted to use the other features of the system to do the purification, plating, and send it to assay, we had that flexibility. And that's something really important to mention with this system. It's not an all or nothing proposition. When I'm going through this process, I showed the complete process, but you can use the system a la carte and really bring in the science in and out at almost any point in the platform. The other thing is once the system brings a rack off the platform, as Ryan alluded to, the track acts as kind of like an Uber Uber system where you have these racks acting as vehicles. And once a rack is taken off of a certain type, we had these carousels that had redundant racks that could be put back onto the track to allow for efficient operation. And then, again, a minimized touch point. The operator only had to refill racks at this stage of the process. Now, something that just came in toward the end of 2019 was an on-demand solvent delivery and solvent dispense and delivery system that was designed by the Morse Group that allowed for us to effectively deliver reagents to all of our modules and also satisfy some safety requirements of the room itself. When the lab was being built, we designed this solvent room where the solvents would be stored in a single location, distributed out to this, where this yellow star is, to this distribution hub, and this distribution hub would then send solvents to each one of the modules. So from an operations point of view, instead of having to manage solvents on 16 different modules, check the level, be changing out solvents frequently, we had a single point for where our solvents would be stored, tracked, and from an operations point of view, be able to be managed much easier. And then also for solvents that needed to be refilled, we had this it really is almost like a beer tap system where you can walk up and operators can fill solvents in their vials and vessels and carboys without having to carry solvents from a dock and so forth. So operationally, this was a really nice feature that we wanted to incorporate. And a little factoid, this is the largest solvent delivery system in the county of San Diego. So again, this really highlights the fact that even though we're doing discovery science, we really wanted to think about the manufacturing and really having a robust platform that could run like a production facility. Now, the other thing to fuel the lab, and this is something that I've worked on now and partnering with some key partners in the reagent industry, enamine and e-molecules being two of them, is this just-in-time flat pricing reagent ordering. And there's a lot of details in this slide, but a couple things to highlight, one of which Pfizer and Millipore Sigma just published on a very similar concept for their parallel medicinal chemistry group. What this does is instead of ordering reagents traditionally by the gram, we order just enough material, and this was highlighted in the video. For many of the building blocks that we use, we only use them one or two times and then have to spend a significant amount of OpEx in storing, tracking, and the most expensive step is disposing of these reagents. So if we can just order what we need and then be able to use that and then not have to, if that product that we make ends up being interesting, we have a supply chain that we can get more quickly. And what this does is there's a number of advantages to this. Now, a high number of our compounds do not need manually reformatting from the vendor-supplied vials into our standard vials, so that's one or two less ways that we have to do by the automation or by a technician. And then as a compound designer, this is one of the things that I absolutely love, is that whether a compound, to make this particular compound, it would cost $42 to buy a gram of each of these, and to buy this compound, it would cost over $1,800. With this flat pricing model, if I wanted to run both of these reactions, it would cost the exact same amount of money. So from a designer point of view, now this actually now becomes a key component in this hypothesis-driven compound design paradigm that we're running now on this platform, where we can now, as a designer, I don't have to settle for a compound that might have suboptimal chemical and physical properties. I can go right after the compound. If I have a good recipe, I can go right after the compound that is the most interesting and answers my hypothesis. And then combined with that, with this being an automated platform, and again, one of the paradigms that is really different than traditional labs, that the chemist who does the ordering, him or her receives the compound, they use it at their hood, and then they make decisions with it. With this type of lab, we have to gain the trust of our submitters that what we're ordering one is the right material, we're getting it within predictable times, and from a scheduling point of view, one of the nice things that eMolecules has been able to provide is this tiering system that allows us to have predictable shipping times. And again, this allows us to be able to make recommendations back to the submitter that if they design a compound, but it's going to take, I don't know, 12 weeks to get that building block there, would you be able to make another compound with this other compound that we can get in a day? Would that still answer your hypothesis? So that's kind of where we're headed toward being able to work with the submitters and be able to maximize and really accelerate that cycle. So once the reagents are on the system, the next thing is that we need to get them, we need to manage them. And the accessioning step and the weighing step, there's an accessioning step including the physical state of the solid, solid or liquid, which I'll show why this is important in a minute, as well as getting a weight. And Hamilton provided a solution for weighing of all the reagents that come on board this platform in our reagent vials. And then finally, we would store these reagents in our storage system, 2 Hamilton Verso stores that are one's at cold temperature and one's at ambient. So once they're on the system, now we can make the compound. And this gets into, personally, my favorite part, since I'm a synthetic chemist, is the reaction preparation. And if you go back to the example that I showed with the ASL, that first step, most of the time, was a highly manual process where you were dependent on a human to set that reaction up. And we really spent a great deal of time in the design phase to look at solutions that had evolved over the 12 years between the ASL design and the L2S2 design to see if there were products out there that could not only handle liquids and stock solutions, but also handle a variety of solids. And we were able to come up with a solution that allowed for us to dispense pretty much every type of solid that you can think of, both into our reaction vials, as well as these small reagent encapsulation vessels, which you saw in the video that we use a laser to be able to seal these, and I'll talk about the advantages of that in a minute. But one of the things that with the ChemSpeed platform, there was an inherent amount of safety that went into this module because, again, we're using a laser, we're dealing with hazardous materials, possibly pyrophoric, so we needed to use the ChemSpeed enclosure that allowed for safe handling of dangerous reagents, possibly pyrophorics, et cetera. But what, again, the Morris Group was able to do was come in and nicely, all the peripherals that were around these modules still maintain the ChemSpeed infrastructure, but design a nice enclosure around the platform for cameras, webcams, ventilation, et cetera, to allow for a nice visually appealing system. So material transfer technology, in the essence of time, I'm going to go through this really quick, but we spent a great deal of time ahead of this module arriving to really define and look at the different types of solids that we wanted to be able to handle, from crystals to hygroscopic solids to free-flowing solids. And what we ended up doing in partnership with ChemSpeed was build this decision tree based on materials, that the material properties and the amounts that you needed dictated the type of solid handling tools. And just to kind of show real quick, I mean, this is one of the sulfonyl chlorides that we looked at, and these were 2 bottles, but they were 2 different lots from the exact same manufacturer. And as you can see, you have 2 completely different physical states. So in a discovery lab, getting parameterization around this was a challenge, but it was something that if we get these high-level parameters right, we were pretty confident that if we would encounter this type of solid on another compound, we would be able to at least use those starting parameters to be able to dispense that solid in an accurate and the right amount. Okay, so real quick, the solid handling tools would add reagents to these pins that were unsealed, and then we saw the video, but real quick, just kind of in slow motion, showing the laser coming in and being able to pinpoint the right height and the ability to seal that material in that pin. And this sealing really accomplishes 3 things. 1, Now, unlike any other platform that does automated synthesis and solid handling, we have the ability to add a solid last to a reaction. So for the chemists that are in the room, sometimes the chemistry will not work unless you add a solid last, and sometimes you can't make that solid into a stock solution. So now we are able to control order of addition. From a quality standpoint, now we're able to take reagents that would have been very difficult to maintain in their traditional commercial bottles, acid chlorides, sulfonyl chlorides, isocyanates, aldehydes, and be able to pin that entire gram bottle if they were only able to get it in gram quantities, pin the entire bottle, and now we have it in a format that can be used not only on the automation, but by the medicinal chemists that need to use that material. And then finally, by having one component of the reaction isolated from the rest of the reaction, the traditional way that we prepped reactions on the automated system in Indianapolis is we chucked everything into the reaction. And in the absence of having a forward-looking scheduler, that reaction could sit for an hour, 2 hours, because that heating resource is being used. With this technology, now we don't break that pin and release that reagent until that incubation resource is available. So as we start thinking about scaling up materials, running, say, 10 or 15 of the same reactions, we're very confident that the 10 reactions will have the identical conversion every time. So this technology, we feel, is a critical component and a differentiation for this lab. Now, once the reaction is complete, we send it over to our reaction execution module. There's a lot of things here that are important to highlight, but a few things. We have 3 different ways of heating reactions. We have our microwave reactors, which microwave reactors have been involved in chemistry now for a good 15, 16 years. We have our pressure reactors that can run a single rack of vials, 15 or 8, in a single time, temperature, and pressure sets of conditions. So if you're running the same sets of conditions, you can do 15 in one shot. And then we have these indigo reactors. And again, looking at this module, you can see all these peripherals. We have chillers, we have pressure boxes, we have tubing. So from a safety point of view, Morse Group came in, did a spectacular job in enclosing and making this thing look really nice. And then on the back end, again, being able to enclose all of the peripherals, we have a really nice looking module. Now, I want to really touch on the indigo reactor real quick, because this is a technology that we co-developed with Techmatic and a company out of Rockford, Illinois, called Accelerated Machining and Design. This was one of the steps in the process that there was not a robust product on the market that could do what we needed to do here in this for this chemistry. The microwave reactors can heat well. The pressure reactors can heat batches of reactions, but to allow for low temperature chemistry, as well as being able to release a solid or a reagent in that encapsulation pin at subambient. So there's a lot of chemistry that you have to add a reagent cold. We now have that capability. And we ended up building, this is serial number 1, serial number 2. So these are the first 2 indigo reactors on the market. Each reactor has 8 lanes, and each individual lane has its own temperature control, and these sleeves that allow for us to be able to intermix the different size vials, so you're not locked into using the same vial in one position. And also from a safety containment point, now if there's a compromise in terms of the reaction, if the reaction would break, we would just need to clean up this insert, not have to deal with cleaning up the entire system. But the other thing is that we're able to, unlike the ASL where we're not able to reflux, we have a full refluxing capability, as well as using pressure to break the pins. It's a little tough to see in this image, but there was a glass pin in here. And what's nice with this reactor is that you can pulverize these pins, and the glass is just completely almost like fiberglass, nice and gone. And then the lidding mechanism, we have the ability to clean the lid between reactions. And also, the other nice thing is, since we don't have to use a crimp cap, we have the ability to add pins, we have the ability to sample, and we have the ability to do reaction monitoring within this reactor, all fully automated. So we're very excited about having this technology on the system. So to go back to the chemistry that I ran to validate chemoprint, this was the same reaction that was carried out when the two ChemSpeed modules were ready to go. We were able to run this chemistry fully automated from reaction prep. We prepared a pin of our palladium catalyst ligand complex, drop it in, crimp the cap, heat it, microwave it, and the LC showed desired product. So in a fully automated way with no human intervention, what used to be a very highly manual step, we were able to prove that we were able to do this in a fully automated way. Okay, so what type of chemistries that we validated so far? In 2019, we were able to validate confidently 5 chemistries, which are shown here. Real difference between the first 3 and the bottom 2 is that the commodity chemical, what's on the arrow, is pinned, so the same reagent that's used in all the amide couplings would be pinned, where for the sulfon-amide reaction and the urea formation, we would be pinning one of the building blocks. So we call this pinning on demand, which the strategy that we employ in pinning commodity chemicals ahead of time versus being able to pin on demand gives us some flexibility, again, a lot of flexibility, but with flexibility, the best way to manage these on the automation is something we're still working on. And then we have plans to introduce at least 15 more reaction types in 2020 as we continue to have confidence in these platforms. Now, once the reaction is complete, we have a crude reaction mixture, we have glass. How am I doing on time? We got about 15 minutes with the round table, so you just want me to continue? We'll continue on. Keep going. Okay, I think the room's empty, so for those that want to stick around, well, I'm almost done. Yeah, we're leaving. So once the reaction's complete, now we have a crude reaction mixture that has broken glass, insoluble material, and other components in it. And this was one of the steps, and I can't remember if Ryan alluded to this, but one of the foundations of this lab is we wanted to utilize as much off-the-shelf automation products as possible. We didn't want to make this system a full custom automation project where in 5 or 6 years, if that automation vendor would walk away and say, hey, we don't want to support this anymore, we would have been in trouble. So one of the things one step in the process that required a fully automated custom solution was this workup step, which Zinsser Analytic was able to work and partner with Lilly. In fact, there's an application note and case study that they're going to be providing at their booth that will describe this process in more detail. But one of the really highlights of this system, it's a 2-sided system where one side does this workup and sample prep, and then the back half does the actual solid phase extraction. This system itself is really nice because what this does is for these compounds that are going on to prep purification is that you've done this pre-cleanup step where you get the compound in a much more purified state before it goes into prep purification, thus having a lower probability of downtime on those prep chromatography systems. And then also for intermediates, if we're able to get a reaction, and again, one of the nice things is with this 2-sided system, we have a shuttle that can move samples between the two systems. Really nice, innovative piece of custom automation. But one of the just to give you an example of an LC of a reaction mixture, we had a crude reaction mixture that came into workup at about 28% purity. And using the method that Mary Luce and her team in Alcobendas was able to optimize manually in Alcobendas, we were able to get this material to 77% purity and a much better weight. Now, this means that if this compound was a final compound, the purification was going to be a lot easier. But if this was an intermediate, this may be now clean enough that I can skip the prep purification step and go right back to the first step in the process to do the next step in a multi-step chemistry synthesis. So this module, even though it was custom, we felt it was critical in our workflow to have this technology part of the workflow and really done right. So we did a lot of going back and forth with Zinsser, and they were a fantastic partner in providing a solution here. Once the other thing with that module is while those fractions are being collected in the solid phase extraction step, We also taken an aliquot out to go to our analytical and characterization module, which was integrated by Biosero. We have Agilent LC mass specs, and for those of you that have worked with Agilent LCs, they're mainly meant for humans to have a level of interactivity, even though there's automation within the module, within the instrument itself. But a fully integrated solution, Biosero was able to come up with a really nice solution here, which I want to highlight in the video. So these would be the 4 fraction LC fractions that came out of SAP4, brought onto the analytical module and put in the hotel, as Ryan showed in the video earlier. But what I really want to highlight, and this is just a minute video, so we'll get through this pretty quick, is that then the Biosero system would pick up this rack, check it in through the 2D barcode reader, and apologies for the lack of an enclosure. This was during the, right as we got the system delivered. But the system, and you'll notice with the PF400 robot here, you notice that it not only has the grippers, it has that little black nub on the back end of the module. But watch what it does here. Pops open, lifts, and draws out that. So just like a cassette deck, for those of you that remember cassettes, when you push the button, the lid opens up. Same type of system here, where the robot was able to successfully open, close the lid, and put that rack into the module, fully automated without the use of a human. Okay, so the SPE decision tree, I'm going to quickly go, I'm not going to really go into this much detail, but with the Versidian software, Versidian, one of our partners that developed the software to handle the purification and the workup and purification, what used to have a number of human review by exception, now we only have a very few situations where we need to have a manual review. Most, if not like 90% of the samples in this workflow can go through a fully automated workflow without a human looking at it. So once the compound has been taken through the workup module, it goes through a preparative chromatography module, again built by Biosero, the software with Versidian to be able to do, based on the data that we collect in the LCs from the SPE, we're able to select a method and be able to run either high or low pH prep. The analytical approach to go really high level is traditionally a lot of high throughput labs. You would run a synthesis and then take that crude reaction mixture, load it right onto a prep purification, and then you'd have your product out. Well, in a manual world, this is fine, but in a fully automated world, doing this introduces a lot of problems in possible need to do a lot of sample transferring, recombination. You have increased evaporation time, and really the most important is that you have system downtime because you may clog your column, et cetera. So just by adding this pre-purification step to this process, we really wanted to get into this 1X analytical strategy, and both Mary Luce and Andreas are going to be giving a poster tomorrow at 2 o'clock, and we'll be able to provide a deeper level of detail on this process. But it's amazing to see how well, as a synthetic chemist, how well this process works in getting the sample clean and really ultimately get to the sample prep step where we can characterize, which gets us into the last chemistry module, again, built by Hamilton. It's a combination of a reformatting module, QC, and characterization. So we have our 33 ml vial that comes out of the prep purification. We have the ability to dry those down in a Hettich Combi Dancer. We have 2 of those on the platform. Once we dry them down, there's a balance on the platform that Hamilton installed that we get a weight. And the weight's going to determine really the fate of a lot of these other things. We're able to collect the NMR sample, a final LC sample. As you saw with the system, we don't have an NMR on the platform, so our NMR samples need to go out to that output module in racks of 96, and they're run, and that NMR data eventually catches up with the rest of the submission. But really the name of the game here, again, idea to data, is to get enough compound made to make a 10 millimole stock solution. So if the compound is 5 mgs or less, we would solubilize that entire 5 mg sample, and then send that to storage. Anything over 5 megs, we would split that sample, still make a 10 millimole stock solution, and then dry down the rest of the sample to have powder in a bottle. And that powder in the bottle could either be sent out to a chemist to do the next step in the synthesis, sent to our storage facility on the system or offline, or it could be an intermediate that we send back to the first chemistry module to do the next step in the synthesis. So now we get into the compound storage, which is the end of the road for me, where now we have an A1 vial in DMSO, a 10 millimole stock in DMSO that is ready to be stored and sent to assay. Now, the overall process time, because this is one question that we get asked a lot, is how long does it take to get through that synthesis process, the make process? If we do a 30-minute microwave reaction, we estimate, based on all the steps that I just went through, it should take about 7.5 hours to get through a compound from reaction prep to a solubilized sample in DMSO. For a reaction that would, say, take 8 hours conventionally, we'd be looking at about double the time. So when you look at, again, annual synthesis reaction capacity per module, we were able to do a high degree of modeling ahead of this system before finalizing the platform. And we were able to determine that our bottleneck in the whole process is our reaction preparation step, which is the very first step, but it's fully automated. And I kind of like to draw a triangle where you have throughput, redundancy, and functionality. And each one of these satisfies one of those three ends of the triangle. But for reaction prep, since that ChemSpeed module and those tools, we didn't have a lot of data around it. We chose the functionality route. We chose all the tools that ChemSpeed was able to offer us, and then we would use that first module as an opportunity to get data on these tools, see which ones were fast, which ones were more accurate, which ones were robust, et cetera. So we could run, get data, but still, we estimate from start to finish, we could run around 20,000 reactions in a year. But a couple of assumptions for those, we're assuming that this is all run on a system that has a fully functional, integrated functioning scheduler. We also modeled in some downtime where we would have a couple months out of the year for maintenance for the entire lab. And for the synthesis suite, we estimated a 24-hour, 6-day-a-week run for 48 weeks a year with downtime throughout the lab. Now, this shows the whole workflow, so which is our simple workflow, but it kind of shows really from start to finish how we feed the system and the output that I just described. But what's nice is this workflow has opportunities to bring in intermediates and other types of work over time. So the ability, again, build this foundation, get this workflow fully automated, validated, being able to do multi-step chemistry and more complex chemistry over time, hopefully should be pretty straightforward. So some final perspectives from a scientist that has been working in automation now for a couple years, really with this project. I'm not going to read these verbatim, but really it's about the cultural barriers, the operational barriers, allowing new technologies to come on board within an organization. Automated synthesis was never intended to replace chemists. It was a tool to increase individual team productivity. There's technology advancements that have emerged since this project started that would, I think, even help this closed discovery loop even more to be filled or to be completed. And then the last thing is that the artificial intelligence machine learning portion of this lab's collecting, as David Evans alluded to in the video, we're not only collecting biological and chemistry data on this system, but the process data that we're collecting, I don't know of another system that is like this, that is collecting as many steps in the process down to reagent quality, lot numbers, et cetera, in what we call events, that every event is tracked on the system. So for those of you that work in AI, machine learning, and want to talk to us about potential partnerships, that data might actually be more valuable than the biological data that we're going to generate on the platform. I know that those are two separate things, but just in my opinion, looking at the system, the process data is just rich, and it's standardized, and it's ready to I think, be ready to be mined by artificial intelligence once we've demonstrated full automation. So I will pass the floor back to Ryan to wrap up the last little section.
I promise you we're almost done. But if we stopped here where Todd left off, we wouldn't have completed that closed loop. We wouldn't have gone from idea to data. And so what I want to talk about is once we have a stock solution, we did calculations and determined that with Lilly's Tier 1 testing, we only needed about 5 milligrams of material to get us through, get us to all the answers that we wanted initially from a product profiling. And so we take a stock solution and we have the ability to take it to a traditional volume reformatting where we can go from one of our A1 vials and take it into assay-ready plates. We can also create master plates. We can go into different types of vials. And then once we're in a state where we're in a master plate or even acoustic dispensing tubes, we can generate assay-ready plates in a low volume, so we have acoustic dispensing module. Our traditional liquid handling volume module was developed by Hamilton Robotics. Our acoustic reformatting module was developed by LabSite. Every compound that's made on the system is going to get an NMR and an LCMS data for characterization. Every compound that's made on the system is going to go through an initial Tier 1 biophysical testing module. If the data looks very promising or interesting at that point, those compounds can then go into biological or cell-based assays. We have the ability to do weekly routine biochemical cell-based assays. We also have the ability to do some batching at this stage and go into more portfolio or target-specific assays. And so this is our biophysical testing module. It was developed by Hamilton Robotics. It gives us the ability to look at our Tier 1 biophysical assays. We also have additional capabilities on this. We understand that Tier 1 is a bit subjective. Lilly has their Tier 1. Other research organizations may have their Tier 1 data. But we're looking at things like high-throughput thermodynamics solubility, high-throughput storage stability, log-D, affinity selection, mass spectrometry. This has the ability to look at 500 to 1,000 different compounds per well. And then using our analytical module to then do final analysis. Our biological testing module was built by Biosero, and it's also has software that's been developed by Stratios as part of that. We're looking at biological assays here, where we're doing biochemical activity and binding assays, as well as some additional cell-based assay capabilities. One of the things I talked about early on is this idea that we wanted to build a platform where we could have integrated workstations or science being run in any other Lilly site around the world or any other research site around the world. And once it was validated and deemed to be working and it would be beneficial, we wanted the ability to then reproduce or relocate that and open those capabilities up on as part of L2S2. A great example of that is as part of our 2020 Phase 5 expansion, we're now doing a biological testing expansion. We took a set of integrated workstations from our Indianapolis facility known as the Advanced Automated Biology Lab. We had over 30 different projects and assays running on that. We've since relocated that from Indianapolis, and now we've broken that into 3 different modules that are going to be integrated or in the process of being integrated onto L2S2, and now those capabilities are opened up to a broader research collaboration. We have the ability to do increased cell-based assays. We have a module that's geared for high-throughput screening, and then we have an additional module that's opening it up to automated transwell culture, some ADME-tox capabilities, and some additional biochemical assays. Again, I want to close the talk by just thanking each one of our collaborators, integration partners, labware vendors, and we have booth numbers on the slide here, but big thanks to Hamilton Storage Technologies, Hamilton Robotics, Zinzer Analytic, LabSite, ChemSpeed, Stratios, Biosero, the Morse Group, Accelerated Machine Design and Engineering, Techmatic, Versidian, and Simplicis. If there's anyone in the audience today that's interested in learning more about accessing L2S2 capabilities for your own scientific needs, I'd like to encourage you to talk with our operational partner, Stratios. I believe we have a Stratios representative in the room, but their booth number is 2116. There's also a contact at the bottom where you can email with questions. But they're really excited about making your own assays available on demand and allowing you to leverage the L2S2 for your own capabilities. Here's an exhibition map. You all probably have that, but these are where these integration partners have been located. And I just want to finally thank all the many people across multiple organizations at Lilly, multiple very diverse areas of science for their contributions. I want to thank the design team here that has been working on this really since mid-2015. Thank many other people here in the audience and elsewhere that were part of this in some capacity. And certainly not last, last but not least is our past and present executive sponsorships for not only believing in this vision, but believing in funding and supporting the vision so that we could turn this into a reality.